Alright guys, so today we'll be discussing about the microbial groups, alright? So we're going to talk about the different infectious agents which are medically important, right? And they're very important for actually causing human diseases, right? So whenever we talk about, let's just say, this group, we'll just say these are microbial agents. We'll talk about that. And the different types of microbial agents that we've discussed about, right? And then one of the categories that we talk about is, all right here, very, very important, something called, all right, we'll make this structure right here. What is this structure right here? This is your virus, right? So we'll just make it a virus, right? So we'll just make it a virus right here like this, right? And this is basically, virus have either RNA or DNA particles or something like this, right? Now, this is a virus, correct? Now let's just write down virus here. So we have to talk about what this is. But not only virus we have to talk about, we have to also talk about this structure right here. And what is this structure is called? That is basically a bacteria, right? So I'll just quickly draw and make the structure, all right? So basically, we'll talk about the structure right here, all right? We'll just make it like this, all right? And then you'll have a basically circular DNA like this, right? And also we'll just make it quickly, all right? It has usually most of the bacteria have let's just say, cell wall. And it has something else coming out from here, right? Like this, right? And this is something called pili or flagella. We'll talk more about it. But this is let's just say your bacteria, all right? And we'll talk about this too. Now, not only that, we also have to talk about some other stuff. And that is, all right, we have to talk about another type of agents, and that is, let's just say, we'll go with this one. This structure you see right here, that is called fungi or fungi, right? So we'll just, if I were to quickly make that too, we'll just make it a structure like this, and then we'll make it a structure like this, right? And then, look at this. This is your, basically a fungi, or fungi, whatever you like to call, right? Now that one. And then third, and then last, we also have to talk about this one. And that is, your basically structure. We'll make this in we'll make this in a red, right? So usually it is what is this? All right? Look at this. And this is your, basically you can think about this as a parasite, all right? So we have to talk about this, parasites. Now, so this is very important. So you have to, whenever we talk about the infectious agent that actually, causes human diseases, we have to discuss about these four ones, right? And there is a concept called prions too. Okay, let me just quickly add that up too, which is now, we'll talk a little bit about, prions also, all right? But now, before we go talk about this, let's describe what viruses are, what bacteria are, what fungi, and what parasites are. Remember, we're going to give a general, this is like general topic about this, right? Because this is we're trying to build the foundations of it. Then after that, we're going to go very detailed about each of this, this microbial agents, right? Now, whenever we talk about the viruses, the viruses are what? You have to ask yourself a questions that, the viruses, do they have cell or not? Are they cellular or not? That you have to ask. And also you have to ask questions about, whether or not the viruses are like organisms, like a living organisms, right? So that is something that you have to remember. Because, viruses, before we go talk about, let's just write down sizes. Usually viruses size they're very, very small. Like it's usually about 0.022 and 0.3 microns, right? So usually virus you can't see through your naked eyes, of course. Even through the light microscope, you can't see, right? So that's something that you have to remember. Now, this is very, very smallest sort of infectious parasites or agents, right? Now, these viruses, do they have cell? Of course not, right? They are not. They are acellular organisms. So you have to remember this, right? On top of that, these viruses, whenever they infect us, right? These guys tend to invade inside the tissue, right? So it's because of that, these are obligate, right? Obligate intracellular organisms, right? And these are intracellular, we can simply say, let's write down parasites or infectious agents, right? Now, you have to remember this, that, viruses are acellular, and this is obligate intracellular organism. That means that, let's just give you examples of this. Like, for example, let's just say viruses has, what do they have? They have either DNA or RNA, right? That's what they have. So they will have either like this, right? They have, this is a maybe single-stranded RNA, or it could have it could be single-stranded like this, or it could be double-stranded RNA, or it could be single-stranded DNA, or it could be double-stranded DNA, right? They'll have that. On top of that, they'll also have basically, what is this black? The protein, right? The protein cover, right? So together, whenever you have this RNA or DNA, or we'll just write or, and you have this basically, this protein cover like this, it has it, we call them nucleocapsid, right? What do we call them? We call them a nucleocapsid. That's all. So but remember, it doesn't have a nucleus though, right? So if you hear the term nucleocapsid, it basically means that either it has RNA or DNA, right? With this capsid or this cover on it. And these covers are made by a protein. Now that is what's called. Now on top of that, you probably have also heard something called, whether these viruses are enveloped viruses or they're naked viruses or not enveloped viruses. What does that mean that? Which basically means this, that if you look at this virus, right, which is a single-stranded RNA, let's actually draw here. If this, let's just say this is a single-stranded DNA with the cover on it, which is called nucleocapsid, right? But on top of that, it also has basically, let's just say, envelope, like with a cell membrane there, right? Cell membrane, usually they steal this from the host, right? And if they have a basically cell membrane, also the outer covering by the, let's just say, lipid layer, right? Then we call them enveloped, all right? And this is called envelope because this is covered with the dress, right? But the dress is what? But what kind of dress is this? There's a lipid dress. And where did they get this lipid dress from? They got it from the host cells. Like it could be, like host would be like, maybe it could be human being, right? Like us. So, but if there is no, let's just say, cover on it, right? That means these are naked because they don't have the sort of the dressing. They don't have this cover on this virus particle, all right? So that is something that you kind of have to know also, right? Now, obviously, the viruses, right, by definitions, we say that it's acellular organisms, right, which basically has a DNA or RNA with the protein cover on it. That's all it means. Now, after this, you also have to remember this. And we also talk about with the virus that can be enveloped or naked also, right? That is something that you have to remember. Now, if you want to remember this, like, do viruses and how do viruses multiply, or how do they cause diseases? Basically, when the viruses hijacks, right? Let's just say if I make this, virus. Let's just I'll make this as a, one, like a virus right here. If this one hijack us, and we'll just make it like, let's just say, I'll make another particles right here too. Like let's say it has a nuclear covering also, right? And we'll make it like double-stranded this time. And it has basically this, something that, we call them a spike, something that can attach. And we'll talk more about this too. Whenever this let's say, invades to the tissue, let's just say, for example, okay, we'll make this as a human being right here, all right? We'll make this as a human being right here, all right? Look at this. What happens if this wants to like hijack you, right? It will go to this cell inside right here, right? And that will stay there, and they use the host cells to actually, go through a process of polycisternic, right? And what they do, the way they tend to do something called polycisternic. Now, what is this whole concept of polycisternic basically means? Remember that we're talking about mRNA, right? The way they're going to synthesize more proteins for them to be more functional and cause diseases, right? So these are polycisternic. Cisternic, like this cisternic referring as genes. It's the old name for genes, and then poly referring to many genes, right? So what happens is that one mRNA can basically encode for multiple or different types of proteins for them to basically be more functional or be active, right? That's what this polycisternic means. It's very, very important that you have to remember. Now, you also have to remember that, okay, the way they do replication is very difficult to understand, right? It's very complex the way they do it. But, and we'll talk more about that when we talk about the viruses, but this is a general topic about it. But now, as I also said that this is obligate intracellular organisms, then if this is obligate, meaning that this is going to hijack inside the cells and then hold this replication process and make another new virus or virion particles, then you have to ask, like, how does the antibodies that works then, right? Because remember, antibodies are what? They're glycoproteins. They do not cross the what? Plasma membrane, correct? So usually antibodies are very, very good for extracellular, but not for the intracellular organisms, right? So then you might have to ask the questions. Then how do we, all the time you hear, let's just say you're getting antibodies, right? Like for example, nowadays you oftentimes hear that, you're getting the antibodies like vaccines for the coronavirus, like COVID-19. So how the vaccines is actually protecting us from the COVID-19? One of the reasons what that is, one of the way the antibody does is that, look, it basically, if there's a virus particle it got inside you, right? Antibodies will go and bind into this like this, outside, right? Before it hijacks the cell, before it invades before it becomes the intracellular, it basically goes and binds to these spike proteins, right? So when that does that, it neutralizes this. So it neutralizes this. One of the functions they do. On top of this, by doing that, they're also preventing the attachment to the host cells. That's also they're doing, right? So these are a couple of reasons. There's a couple of reasons why you should get vaccinated, and this is how it actually prevents from causing disease, right? So that is important to know. But now, for you, remember, viruses, acellular, it's obligate intracellular, right? And either it has a DNA or RNA, and we also talk about nucleocapsid. These are polycisternic, right? You have to remember this. And do they have let's just say, ribosomes? They don't, right? They don't have their own ribosomes. So that's something that you have to remember. So they basically use the host cells for everything in order to make new virion, right? That is why usually outside of human being, these are not living organisms, all right? Now, after the virus, right? And we'll talk about different types of viruses, right? Like for example, influenza virus, coronavirus, hepatitis B virus, right? Polio virus, you'll have a cytomegalovirus, herpes viruses. We'll talk all of them in the virus topic. Now, after this, let's talk about the bacteria now. Remember, bacteria, generally, what do we call them back? We call them prokaryotes, right? Bacteria are called prokaryotes. And usually fungi and parasites are called eukaryotes. Now that means that you have to also know the difference between the prokaryotes and then eukaryotes, right? Something that you remember. Remember, this is a bacteria right here, right? This is your basically a parasites, and this is your fungi, right? Now this fungi and then your parasites, these are what? These are eukaryotes, like us. But bacteria are prokaryotes. Now what does this mean then? Usually, whenever we say they are prokaryotes, what do we mean? But before we go, what is the usually size, the average size of bacteria? And if you want to remember, usually it's like, okay, you go from 0.2 to basically, 2 microns, right? So if you remember this, that's usually the size of the bacteria. If you remember the fungi bacteria, or like fungi, or parasites, let's just say, it can go from maybe like 15 to like, 25, sort of a micron. Right? Or if you remember, let's just say, from this guy, fungi, it's like 2 to, let's just say, 10 micron. Right? Fungi size of this. Now, these are usually the size of this. So bacteria is usually larger compared to like, the virus. Right? Now, you have to remember this, that the bacteria, they're prokaryotes, which means that, this almost all the bacteria have something called cell wall. That's something that you remember. But I said almost all the bacteria, which means the majority of the bacteria. But obviously there are exceptions. Medically, you should remember one bacteria, and that is called mycoplasm, all right? That mycoplasm do not have a cell wall, all right? Do not have a cell wall, and that is very important for you to remember, right? Now, on top of this, if you talk about, let's just say, in the bacteria, do they have a DNA or RNA? They actually have both, right? So they contain both DNA and RNA, just like, even the eukaryotes, they have both DNA and RNA, right? Both of them have that. But you also have to remember that usually in the bacteria, right, the DNAs are more in like circular fashion, like this, right? But here is more like a linear fashion. In the eukaryotic cells, they're more like a, we'll have to say this is more like a linear fashion, just like a human being, all right? But this is more circular. That's something that you remember. Now, third point you should remember here is that, bacteria they're non-membrane bound, all right? Or membrane, they're not, they're non-membrane bound with organelles, right? That is something that you also remember. This is membrane bound organelles, right? These are membrane bound. Oh, look, okay, organelles, right? This is something that you have to remember. Now, fourth point you should remember is that, remember that the bacteria, whenever they do a transcriptions or translation process, right, that happens, both the transcription, let's just say, both transcriptions, right, and also like translations, that takes place primarily in what? In basically, cytoplasm. But if you talk about the eukaryotic cells, right, what is happening here is that the transcriptions does not take place in cytoplasm, instead it takes place in a nucleus, right, but then translations, right, takes place in a cytoplasm. Very, very key point that you must remember this. Now, fifth point you should remember is that, these do not have a nucleus, prokaryotes, but they have a eukaryotic. We do have a nucleus, right? And nucleoli. Bacteria DNA or RNA, we call them a nucleoloid, right? That's what we call them. So these are called basically nucleoid, actually, we call them a nucleoid. So look, general concept, prokaryotes, they have a cell wall. They have a DNA, they contain both DNA and RNA, right? But even though they contain both DNA and RNA, right? But usually there is no histones are present, right? It's very, very important. But here, the histones are present, right? So this is like some of the facts, but you should remember this. Now, on top of this, whenever they multiply, right? They go from monocisternic to polycisternic. So this is something that you also have to remember. And so polycisternic basically referring as what? Cistern is referring as a gene, the old name for genes, and poly means many. So it means one mRNA can encode for different types of proteins in order for basically, for to do their function. Like for example, let me give you examples of this bacteria called E. coli, right? E. coli use, they can use something called, like, let's just say, they can do something called lactose metabolisms, right? They do something called operon. What is operon? Operon basically means that, it's a gene, they encode, they have a, basically the operon is they can operate this basically polycisternic gene, right? So lac is referring as a lactose. So, the reason why we call them a polycisternic is because this one mRNA, right, that can encode for different types of protein in order for the lactose to get metabolized. That's why we call them a polycisternic, all right? But if you're talking about the eukaryotic, like fungi and parasites, they are monocisternic, right? Because one mRNA can only, let's just say one mRNA can only encode for one basically protein. So they are monocisternic. This is something that you have to remember. Now, it's very, and whenever we talk about, let's just say bacteria, right? The way the bacteria, let's just say, multiply, when the bacteria multiply more or in a continuous fashion, right? They're always multiplying. And also you have to remember something called whole concept of a binary fissions, asexual reproductions. I will talk more about this. But if you talk about this guy right here, they usually primarily multiply, this takes place more in a G1 in the cell cycle, if you have studied this, right? It takes place replications and all, it basically takes in G1 and S phase. But this is more like continuous, right? And this multiplies by binary fissions. What does that mean? Which means that, all right, look, if I quickly make this, I'm going to make a diagram, right? And this is something that you have to know. If you've seen a graph like this is a, if you basically, let's just say, inoculate some bacteria and put it into the culture, right? This is what happens. This is a log of the bacteria, right? Bacteria. And you probably have seen graph, and this is like time. Right, so usually what happens is that, let's just say you inoculate this bacteria, like let's just say, this bacteria, right? What happens? There's one, right? So this is one right here. But when you inoculate this in culture, right? You'll see something like this. And then after that, what happens? This was studied like, maybe this time they're using nutrients, right? So it's because of this, or, they're producing, they're basically, going through the process of, they haven't started replicating, but they're using all the nutrient sources for them to actually start getting, multiplying. So that's what happens during this phase, right? So that's why you see this sort of like a graph, sort of a linear graph, right? Then after what happens is that they started multiplying. And when they multiply, look, this is a 1 right here, right? They started multiply 1 and it become 2 like this, right? And after that, they'll further multiply, this become this, right? And this become this become like, let's just say, so it basically, 2, it becomes 2, let's just say like this. Okay, here. 2 right here. So it becomes 1, become 2, 2 become 4, 4 will become basically, let's just say, you can say it will become 8, right? And then 16 and 32, that is how they multiply. The way they are growing now, it's called exponential growth, right? This is a logarithm growth like this. It's going to go, this is exponential growth. This is a logarithm growth, because this is the time, the bacteria are actually multiplying, right? And they multiply basically, binary fissions, right? 2 gives 4, 4 gives 6, 4 gives 8, 8 gives 16. That's how they basically multiply. And this is this is called the bacterial, graph, right? Growth graph. And this phase you see, right? This phase is called what? Accelerating phase. This is called exponential growth phase, right? This is multiplication phase, right? These are like binary fission phase. That's what's happening, right? But this is multiplication phase that's happening, right? And there's a rapidly growing. But one thing that I want you to remember is this phase right here, right? Most commonly we call them exponential growth phase or logarithm phase, all right? So that's something that you remember. And then after that, what happens is that they start getting a plateau like this, right? There's a plateau. Now what is the concept of all this plateau thing too? Talk about that. And then after they start falling down like this. Now we'll talk about, and this is the time it takes to do, right? So now, all the way down from here, when the bacteria was, let's just say you inoculate some bacteria and put in the cultures, in the right away, let's just say maybe like this is like 1 hour, we can say this is, let's just say 1 hour, 2 hours, 3 hours, like this, So basically you can think of it this way, that maybe during like the 1 hour, some, different types of bacteria, depends on what type of bacteria, they have, this time frame is not important. Just understand the concept right here, right? So what really happens is that some of them grow quicker, some of them multiply faster, some of them take a little longer time. It also depends on the kind of nutrients and the growth factor that is actually present for the bacteria to have sort of exponential growth. So basically, this phase right here, when the bacteria is basically not multiplying, they're not going process of binary fissions. This phase right here, right? This phase is called, we call them a lag phase, right? In the lag phase, basically the bacteria are probably using nutrients products, right? They're basically maybe getting swollen up, they're eating up, right? That's what they're happening here. So, but the numbers of the bacteria remains the same, all right? They do not multiply. But then after that, what happens when bacteria started multiplying, this is called exponential phase, exponential growth phase, or logarithm phase. This is a very, very important phase, because in this phase, this is what is happening. Bacteria are multiplying rapidly. That's why you see exponential growth, right? And the most important for you to remember here is pharmacologically, antibiotics works best if you give on the exponential phase. All right, it is very important there. Now, when they are multiplying so rapidly, so growing up, right, what happens is that eventually what happens, they'll reach this phase, which we call them a stationary phase. This is called, let's just say, stationary phase. Now, what is this whole concept of stationary phases? In this case, look, when these bacteria are multiplying like this, right, the bacteria needs what? Nutrients. They need growth factors, right, for them to survive and do more functions. But really what happens in the stationary phase is that, maybe like some of the bacteria are, because there's so many, the nutrients or growth factors, right? Those are started becoming what? Less, right? It's because of that, some bacteria are started dying. Right, that's what's happening. On top of that, the toxic metabolites that started getting accumulated there, right? And that also destroys bacteria. So basically in the stationary phase, numbers of bacteria that multiplies equals to the number of bacteria that are dying. Okay, it's because of that this become plateau, right? So numbers of cells that is multiplying is equal to the numbers of cells, or numbers of bacteria that are dying, right? That's what happens in the stationary phase. That's why you see this graph, which is more like a straight graph, right? Or plateau, we call them, right? And that is called stationary phase, right? So then after what happens is that, bacteria die, there's so many bacteria gets accumulates, there's so much of toxic metabolites are there. So what started happening is that there will be the more cells, the more bacteria start dying. And when you see this, you start seeing the decline on the bacteria multiplication. So that's why you see this graph. This is like a declining phase, right? So bacteria are dying, and this shows that bacteria are basically dying. And then all the way comes to down there. So this is very, very important. Right? You should at least have an idea of this. Okay? Now, bacteria, as I said, right? They have a cell wall, they have a DNA and RNA, but without stones, they are not membrane-bound organelles. That's something that you remember, right? They're both transcription and translation that takes place in cytoplasm. They don't have a nucleus. They're polycisternic, right? And the way they multiply is basically the binary presence, right? And now, on top of this, you have to remember something very, very important, the concept called ribosomes, right? It's very, very important. Now, what is the whole concept of ribosomes is this, that, they have something called ribosomes. But these ribosomes are a little different ribosomes. And what type of ribosomes is this? Let's just say these are ribosomes, right? And let's just say this is a gene right here. Because ribosomes are very, very important, right? For basically helping synthesizing a protein, making enzymes and functional proteins. Now, the ribosomes that are actually present in the prokaryotic cells, right, in the bacteria, you must remember this. These are what? These are odd numbers. They're 30S plus 50S, right? Together, when you add them, does it become 80? Of course not. It becomes 70S, right? This is very, very important, because why it's very important for you to remember is that there's so much the drugs that works there, right? Pharmacological agent. There are drugs like let's say 50S inhibitors are there, right? Or there are 30S inhibitor drugs are there, right? So you must remember this. Like for example, if I have to give you an example like 50S inhibitors, have you ever heard something called macrolides? Erythromycin, drugs like that works on these 50S inhibitors, right? And if you want to talk about 30S inhibitors, like drugs like for example, aminoglycosides, like gentamicin, or even tetracyclines or doxycyclines, those drugs works there, right, in this protein. So it's because of this, they use something called 30S and 50S together that makes a 70S, right? Now, this is a ribosomes, okay, of this, what, bacteria. So it's very important. These ribosomes are very, very important for what? Synthesizing proteins. So please, you must remember this two, 30S and 50S, very, very important. Because there is a pharmacological drugs is associated with this. So very important that you must remember this, okay, about the bacteria, all right? So after this, let's talk about, let's just say eukaryotic cells. Remember, eukaryotic cells, we're talking about the fungi and parasites, right? But before we go and talk about it, let me quickly ask you something. The prokaryotic cells, we say, that here, we say that their cells, the DNAs are circular, right? And then here we say the eukaryotic cells, the DNAs are usually linear, just like our cells, right? We have a linear DNA. But do we have a circular DNA? Let's ask a question. Do we have that? And if you say the answer is yes, then you're absolutely right, okay? Because think about it. Let's just think about it. Why do we find the circular DNA in our human body. And if you say mitochondria, you're correct. Mitochondria, right, even though in our cells, in a nuclear DNA, we have are basically, they're linear. But mitochondrial DNA, right, the mitochondria, which everybody knows as the powerhouse of the cells, ATP producing organelle, right, it has what? It has a circular DNA. Now, that, that reminds me of something. Long time ago, billions of years ago, what really happened is this, that, this, let's just say this one bacteria, right? What happened was this, let's just say, I'll make it a bacteria down here. We'll just make this as a one bacteria like this, right? This one big prokaryotic cells, they engulf the another prokaryotic cells or another bacteria, right? That's what really happens, like this. So now, let's just say, for example, this is a bacteria, right? But now what happens is this, that this bacteria, right, engulfed this bacteria. Now it's here, right? The bigger prokaryotic cells, right, prokaryotic, engulf the another smaller prokaryotic, right? Now, when this happens, this smaller prokaryotic, right, that did not get digested, right? Instead of breaking down process, right, that did not happen. So breakdowns of these small prokaryotic cells or bacteria, that did not happen. Instead, they started forming a very good relationship between them. We call them symbiotic relationship, right? Meaning that what is a symbiotic relationship basically means? That, they started making such a beautiful relationship where that, they both are sort of like, started becoming benefits with one another, right? That's what it happens. So there is a good, beautiful relationship they sort of like made, right? So it's because of this, and this has happened like billions of years ago, right? And somehow, this agent, they got invaded to our human cells also, all right? So when they got this in our cells, right? Basically because we cannot, we were not able to break this down because of the symbiotic relationship, what happens is that, they are very, very important. They started producing something called energy. This prokaryotic cells, right, started producing basically energy, ATP for us, right? And now, right, we don't call them a bacteria producing ATP, but instead we call them a what? Mitochondria, all right? So this is how. So now, next time if you have mitochondria, it is basically a bacteria who's producing energy for us, which is basically what? Pumping us up, right? So just remember that, and I just wanted to give a little bit of a, sort of a background of like this whole, the concept of mitochondria, right? Rather than just saying it's a powerhouse of the cell. Now after this, let's talk about now the fungi and parasites because that's important, right? Now, whenever we talk about the fungi and parasites, obviously they have DNA and RNA, right? Obviously they have histones, right? They are membrane-bound organelles. Transcription takes place in nucleus. Translation takes place in cytoplasms, right? And when they synthesize protein, they are monosystemic. You must remember this, right? And whenever we talk about ribosomes, right? They have basic what? Something called 40S plus 60S, which basically doesn't make 100, it makes 80S, right? It's very, very important. The ribosomes, ribosomes are 40S and 60S in order to synthesize the protein. That's a very important concept also. Now, on top of this, you also remember this, that, the cell membrane, even though, look, bacteria has a cell membrane too. We didn't talk about that, but we'll just write down here. The cell membrane, right? But usually the cell membranes are, obviously there are exceptions, but they are, they're usually phospholipids. Or we'll just say, but there are sometimes the lipooligosaccharides too. But for simplicity, we'll just say they're lipid. They're lipid-bound membranes you usually see in the bacteria, right? When you talk about, let's just say, in the fungi, right? what do you see? You have to remember this, that fungi also have a cell wall, right? Now, the cell wall is, you have to remember, but cell membranes are usually orgosterol, right? The orgosterol, when you're talking about, let's just say, parasites, cell membrane they use is cholesterol, or just like us, human being, right? But they also have a cell wall, right? And the cell wall in the fungi are used by something called chitin, right? And here, usually in this parasite, there's no cell wall. Okay, so you must remember that fungi, they have a cell membrane that is made by orgosterol. And it's very, very important that you have to remember this, because orgosterol, there is a drug that actually works on the orgosterol, and that creates pores, all right? And that is how we destroy this fungi. And the drug that actually goes and binds to the orgosterol is like drugs like, for example, anistan, niacin, or amphotericin B. We'll talk about that. And then there are drugs like cell wall too, right? There are something called micafusine or micafusine that actually goes and works on the cell wall too, right? So it is very, very important that you should remember both of this. So very, very important. This orgosterol, and for the chitin, you must remember for the fungi, right? Parasites, usually drugs, usually the cell membranes, or what is this called? They have something called cholesterol, right? So very, very important. Now, in general, right? In generally, we talked about, what viruses are, what bacteria are, what fungi are, what parasites are, right? And these are basic, look, these are examples of bacteria here, look. This is and bacteria have different shapes. We'll talk about it, right? This is viruses. And this is, let's just say, parasites. Like, for example, like Treponema pallidum, or, like helminth, sorry. So that's one, right? Parasites. And then this is basically your fungi. And different types of fungi are there, like mucor or even histoplasmosis. We'll talk about all of them, but this is just a general concept about it. Now, after we ended, we have to talk about what about these prions, right? Remember, these prions are neither DNA or RNA. These are basically infectious protein. Usually that these are infectious proteins, right? Which are usually like, which gets deposited extracellularly, and that's how, it can basically, cause the diseases, right? This is something about the prions. So if you ever heard something called prions, you should always remember these are neither DNA or RNA, they're polycisternic, right? And they're polycisternic, sorry, not polycisternic, they're basically infectious proteins. They get deposited into extracellularly to cause basically a problem. They're kind of like, they're more like a, they're more of like, amyloidosis. You know how the amyloidosis, they get deposited extracellularly, and that is very important for causing like problems. So that's how the prion functions also. Now, I'll tell you one more thing, that, whenever we talk about, let's just say the eukaryotic cells, right? If you look at the genetics of the eukaryotic cells, they have we have something called exons and introns, right? And we do something called alternative splicing or spliceomes in order to remove those introns. But if you talk about the prokaryotics, they don't have introns, they only have exons. So this is also something. So that means that they do not go a process of something called spliceomes or alternative splicing. They don't have to, because they don't have introns, but they do. But this one have exons and introns. Right? So the key points for you is that make sure you remember the bacteria have cell wall, except, right? One of the exceptions is the mycoplasm. You must remember that, the exceptions, because they test you. That is why. And the reason why I'm emphasizing on the cell wall is because there are a lot of drugs that works on the cell wall. Like, for example, penicillin, amoxicillin, or ampicillin. Those are beta-lactam drugs, all right? Or cephalosporin. Those are beta-lactam drugs, right? And those beta-lactams are very, very important for what? Part of a cell wall, right? So if there are drugs, if the bacteria do not even have a cell wall, then giving a penicillin or beta-lactam drugs, that's not going to work, right? Like, for example, for mycoplasm, which doesn't even have a cell wall, you can't give beta-lactam or penicillin because it's not going to work. Instead, you have to give a drug more of like protein-S inhibitors, like, the 30S or maybe 50S inhibitors. And most commonly, they give one of those protein inhibitor drugs, to actually, kill the microorganisms, mycoplasms. It's very important that, and we'll talk more about it too we'll discuss more about it too, right? That when we talk about all this sort of diseases. Now, but please don't get confused with the mycoplasm with the, Mycobacterium tuberculosis. That's completely, we'll talk more about that. That's completely separate thing, all right? Now, after this, guys, about this general microbial agents, we'll just end it here. And next class, we're going to talk about the structures of the bacteria. Thank you.